Chapter 8, Applications of Recombinant DNA Technology. Here are the learning objectives for this topic. Describe genetic mapping and genomics and explain their usefulness. Describe 6 potential medical applications of recombinant DNA technology. Describe the steps and uses of genetic fingerprinting. Define gene therapy. Identify 6 agricultural applications of recombinant DNA technology. So in this topic, we're going to go over genetic mapping, environmental studies, pharmaceutical and therapeutic applications, and agricultural applications. Let's go over genetic mapping. Genomics is the science of locating genes on the chromosome. Well, you may ask, why do we want to do this? Well, if you know where the gene is, then you can study it better. Also, when you're looking at human diseases that are genetically caused, oftentimes we can tag down the offending gene to the chromosome, but we don't know exactly which gene it is. So genomics helps us locate the genes that we're looking for on the chromosome. Now, it used to be very labor intensive up until about 1970. We would use high frequency recombinant gene transfer. We would look at crossing over frequency, so we'd have to take the organisms and breed them and see what the phenotypes of the children were and what frequency. We would take a look at restriction enzymes, run gel electrophoresis, etc. And with all of that, we were able to get a map of the gene of Haemophilus influenzae, which is a bacterium with a very small genome. There's no hope of ever figuring out the human genome. Well, then we came up with nucleotide sequencing. That's where nucleotides are selectively cleaved off of DNA. You do gel electrophoresis, mapping order, etc. Each base has a fluorescent tag. We use cDNA for this process. And we get a graph that looks like this, which is similar to what was used in the Human Genome Project, in which we were able to map the entire human genome in far less time than we were planning on. So here we've got different colors assigned to the different nucleotides. This green that looks pretty bluish is adenine, red is thymine, G is yellow, C is blue. And we take a look at this graph and we can map out what the nucleotide sequence is. So here we've got a green peak, that's A, we've got a red peak, that's T, and you could probably extend that on out. We also use recombinant DNA technology for environmental studies. First off, most of the microbes, bacteria, protozoa, etc., in environmental samples or clinical samples are not currently culturable. Right now, we can culture 10%. So there's 90% that we simply cannot grow in the lab. So we have to use recombinant DNA technology. So here's an example of a situation in which we were able to do that. Methane is released by rice paddies. So you can see here's a beautiful picture actually of multiple rice fields, and there's the hut of the farmer. And methane is a gas that is contributing to global warming. Well, I'm not going to go in and tell this farmer that he can't grow the crop that grows best in his region. That's basically telling him that his family can starve to death. Well, I'm not going to do it. By using recombinant DNA technology, researchers were able to find that the rice is not what's releasing the methane. There is an archaea that lives in the rice paddies, and they live in association with the roots of the rice plant. And they feed off of the carbohydrates that are released by rice plant roots and release methane in the process. Well, now that we know what the culprit is, we can study the relationship between the archaea and the rice plants and see if we can work at ways to reduce methane release without telling the farmer to switch to another crop. Let's go on to pharmaceutical and therapeutic applications. Protein synthesis. There are all sorts of proteins that we can synthesize now because of recombinant DNA technology. I've talked about insulin. We've also talked about human growth hormone. Interferon is a big one. They're doing a lot of research on using interferon as a cancer treatment that is easier on the patient than other chemotherapy treatments. Vaccines. Now, the first vaccines ever made were basically domesticated viruses. We call them attenuated. And basically, we've grown them in the lab for so long that they have a very low capacity to cause disease. So we can inject them into you, your immune system sees them, and you develop an immune response. But there's a certain amount of hazard, especially if somebody has a reduced immune system. So we've gone to inactivated vaccines for a lot of vaccines, where basically we take the domesticated virus, the attenuated virus, and we kill it. We inactivate it. Well, there's still some hazards with that. So using recombinant DNA technology, we can make subunit vaccines. That's where we take just the part that your immune system responds to, put it on a non-pathogenic microbe, get it to grow a lot of those antigens, that's the protein that your immune system responds to, and we can make a vaccine for it. Also, we can make subunit vaccines of non-culturable pathogens. To get attenuated and inactivated vaccines, you got to be able to grow the microbe in the lab. Genetic screening. So we can see if you've got a genetic predisposition to certain diseases. Now, that makes a lot of people nervous because, insurance companies saying there's pre-existing conditions. Also, you can do genetic screening on the type of hepatitis C virus or HIV present in a patient and determine what serotypes there are. You can take a look at the HIV and see if it's resistant to certain anti-HIV drugs and use different drugs. Depending upon what serotype of hep C you have, your prognosis is different. Depending upon the serotype, you're more or less likely to develop liver cancer. Helps with the treatment of the patient. DNA fingerprinting, you can use it to ID pathogens, you can use it to identify environmental organisms, and you can identify the perpetrator at a crime scene. We use DNA fingerprinting all the time. To do DNA fingerprinting, you do PCR, then you use restriction enzymes to chop up the copies that you've made, then you do gel electrophoresis, and you get that lovely pattern that we call a DNA fingerprint. Gene therapy. Using recombinant DNA techniques, we've been able to replace missing or defective genes. So far, we've been successful in curing XSCID, or X-linked severe combined immunodeficiency disease. This is because it's caused by one gene that doesn't function to produce one enzyme that's needed mainly by the immune system. So folks who have XSCID are born basically without an immune system. So we've been able to take the gene that doesn't have the mutation and using a retrovirus, use that as a vector to deliver the gene to the patient's bone marrow. Well, the first time that they did it, 5 of the 20 patients developed leukemia from insertional mutagenesis by the retroviral vector. So they shut down the project, cured most of the patients of the leukemia, and then they had to start over. And so far, from what I've been able to see in the literature, we've been able to take care of this insertional mutagenesis so that it doesn't happen anymore. Next, medical diagnosis. We're using an awful lot of the applications of recombinant DNA technology to do medical diagnosis. We use PCR, fluorescent genetic probes, DNA microarrays have become big. We're moving toward what they call bedside diagnosis. What they want to be able to do is you come into the ER, you come into your general practitioner, and you say, I've got a sore throat. And they take a little bit of your saliva, and they run it through the machine, and they're able to say, okay, yes, you've got strep, but it's resistant to penicillin, and you're allergic to sulfa, we can tell from your genetics, so we're going to give you erythromycin instead. The idea of rapid diagnosis, then we can begin treatment, because currently we can do a rapid test for strep because of recombinant DNA technology, but we have to grow it in the lab to figure out what antibiotics it's resistant to. So if we could do a test to see what plasmids it's carrying, speeds things up considerably. Xenotransplants. Xenotransplants are basically taking an organ from a non-human animal and using it in human transplants. Well, the problem with this is immune rejection. So what we do is we insert human genes into an animal so that the patient doesn't have to take immunosuppressives, and they also don't have to wait forever for a human to die and make an organ available. Let's move on to agricultural applications. First one, transgenic organisms. These are organisms that are recombinant animals or plants that have been altered for a specific purpose by the addition of genes from another organism. Currently, the popular name for them are genetically modified organisms, or GMOs. So let's talk about how we can use transgenic organisms in agricultural applications. First one, herbicide resistance. The most common, shall we say, most popular herbicide is Roundup. Well, they took a look in the fields that had a lot of Roundup applied, and they found a bacterium, agrobacterium, that had developed resistance to Roundup. Well, they took the gene from the agrobacterium and put it into crops. Now, in this area, there's a lot of sugar beets that are being grown that are what they call Roundup ready. So you can spray your fields with Roundup. You don't have to worry about where it ends up. Your sugar beets live. Hopefully all the weeds die. Salt tolerance. Unfortunately, irrigation, especially in low-lying countries, leads to a buildup of salt. You take water, you pump it onto your fields, it evaporates, you can see how the salt would get left behind. Well, what they've done is they've taken the genes for salt tolerance out of marsh plants, plants that are living in marshy, brackish, salty water anyway, and they're putting them into crops that people can eat, such as tomato and canola. This has been especially a big benefit in areas like Bangladesh. And not only does it make it so that there's more land available for agriculture, for feeding people, but it also helps remove the salt from the soil. Freeze resistance. There are certain crops that don't put up well with being frozen. Strawberries come to mind. Well, these ice crystals form when they hit certain proteins that acts as a seed for the crystal formation. Well, there is a bacterium, Pseudomonas, that produces a natural antifreeze when we put the gene for that natural antifreeze into the Pseudomonas. Now, Pseudomonas is a normal environmental bacterium. We take these genetically modified Pseudomonas and we spray them onto the crops if we know that there's going to be a cold snap, and we can prevent crop loss. And you're eating Pseudomonas all the time, it's not going to hurt you. Pest resistance is next up. There is an organism, a bacterium called Bacillus thuringiensis, and it produces a toxin that we call the Bt toxin. And when caterpillars eat the Bt toxin, the toxin changes shape in their alkaline stomachs, and it becomes like little shards of glass, and it kills the caterpillars. Now, we can eat Bt toxin because our stomachs are acidic. Those little shards of glass don't form, and it doesn't bother us. So, for years, we've been isolating Bt toxin from cultures of B. thuringiensis and spraying it on crops as an organic pesticide. Well, now they've taken the Bt toxin gene and put it in the crops so that the plant leaves naturally produce the Bt toxin. Kind of cuts out the middleman there. Also, potatoes, we want to make them resistant to a pathogen, a protozoa, called Phytophthora infestans. This is the organism that caused the great potato blight of the 1800s, killed about 2 million Irish in a famine, and had even more Irish leaving Ireland and coming to the United States and other areas, including the ancestors of John F. Kennedy. So what we do is we take and look in other strains of potatoes that aren't quite as nummy, to be perfectly honest, and we take the gene for resistance and put it into the strains of potatoes that we do like to eat. Next up, improvements in nutritional value and yield. We like vine-ripened tomatoes, but tomatoes, once they're vine-ripened, a gene kicks in that causes degradation of the proteins that lead to that nice firm fruit. So in other words, they get mushy. This is a part of the normal life cycle of the tomato, but it makes it so that they're very hard to ship. So in the past, we have picked tomatoes green and let them ripen on the way to the store. Well, you all know how that tastes. That tastes like plastic to me. I don't know about you guys. Well, what they've done is they've taken a strain of tomatoes that they call McGregor tomatoes, and they've taken a gene that produces an antisense RNA for this protein that causes the tomato to go mushy. So when that gene gets turned on, this antisense gene gets turned on too, and the antisense mRNA attaches to the mRNA transcript for the degradation protein and stops the degradation of the tomatoes. And so we can get vine-ripened tomatoes in the stores. Bacterial nitrogen fixers. As you may or may not know, there are certain bacteria that live in association with legumes. They form little nodules on the roots of the plant, and they fix nitrogen for the plant, and the plant provides sugars and protects the bacteria from oxygen. Well, it would be nice if we could get these bacterial nitrogen fixers to live in association with other plants, and we're currently working on that, so that we don't have to apply so much fertilizer. It's all to the good. Okay, last one on nutritional improvements and increasing yield. Golden rice. There has been a real problem in areas where rice is the main food item with not having enough vitamin A. And this leads to malnutrition and blindness. And providing vitamin A is the best way to prevent blindness in children in these countries. Well, the problem is when you take off the bran from regular rice, you remove vitamin A and B vitamins and all sorts of good stuff. Well, the reason why they remove it is because the bran is full of oils, and the oils make the rice go rancid. If you've ever tried to keep brown rice for any length of time, you know what I'm talking about. So what they did is they took beta-carotene from carrots, which is converted into vitamin A in your body, and they put it in the endosperm instead of in the bran. And it makes it this lovely golden color, and it prevents blindness, morbidity, which is disease, and mortality, which is death. Well, there's a problem. It makes the rice taste like carrots, and oftentimes the little kids don't like it. There's also been a lot of brouhaha over golden rice and whether it's ethical and whether they're really cheating the farmers, but we'll get into that in the ethical section. So that's it for this topic. Here are reminders of what you need to get from this topic.